Vibrio bacteriovorus. The living antibiotic. So when we look at Vibrio bacteriovorus, or BV for short, it's a member of the family Delta Proteobacteria. It's very small and about the quarter of the size of E. coli. It's a rod-shaped gram-negative bacterium that is typically an obligate aerobe. It also has high levels of motility due to a flagellum that propels the bacteria forward and allows it to travel as fast as 160 micrometers per second. The bacteria's form influences its function, as they are predatory bacteria that must move quickly to find, infect, and kill their prey, which are other gram-negative bacteria that they attack specifically. Vibrio, the genus, comes from the Greek word dela, which literally translated means leech. An appropriate name for a lob-shaped predator, huh? So, how exactly do these bacteria invade and kill other gram-negative bacteria? As an overview, BV insert themselves via periplasmic invasion between the distinct layers of the gram-negative cell wall and begin to grow inside, lengthening from either end until they divide into multiple daughter cells, which then develop and are released via lysis of the host cell. This occurs over a 3 to 4 hour predatory cycle, beginning with the localization of prey, where a combination of the stochastic movement of the extremely motile BV, along with the fluid dynamics and corresponding settling of target bacterial colonies, ultimately result in their random collision. Once collided, the BV begins to degrade the prey cell membrane using localized peptidase that's on the proximal outer membrane to create a pore for entry. The cell wall through the pore into the periplasm, sealing off the cell wall behind it, and in the process detaching from its flagellum upon entry. The bacterium then establishes itself within the periplasm by attaching to the cytoplasmic membrane and beginning to degrade the peptidoglycan wall using de novo synthesized hydrolytic enzymes, murine hydrolase, and various other glycanases. The BV and host complex is then called a delaplast, and BV grows in a filamentous fashion from either end, replicating its DNA within the periplasm by capitalizing on the host cell's cytoplasmic nutrients. When these nutrients are depleted, the filamentous BV forms into individual cells, which then develop flagella and begin to be motile within the delaplast. Finally, the remaining membrane is degraded completely, lysing the host cell, and therefore releasing the BV cells into the local environment to go on and infect another prey. So, why are these bacteria important? Well, it's basically a living antibiotic that has the ability to kill pathogenic gram-negative bacteria, and we may be able to capitalize on the bacterium itself or the enzyme it encodes and releases as a solution to the growing global problem of antibiotic resistance. One of these possible applications is in patients with cystic fibrosis, a genetic disease that often results in the colonization of resistant lung infections such as Pseudomonas that are hard to treat. But this is a gram-negative bacteria, and in vitro BV has been shown to be effective against it. So aerosolized BV could be a possible treatment for Pseudomonas infections in CF lungs. BV also has the ability to invade biofilms, which is another reason why we think it would be so good for CF treatment, where the lungs are highly colonized with polymeric bacteria-filled matrices that are penetrable by BV, but are not penetrable by normal antibiotics that are typically used. This also makes BV super useful for infections in the mouth, such as periodontitis, since it's able to penetrate the dental biofilm to treat the bacteria itself. BV also has applications in agricultural, animal, and food industries to prevent instances of foodborne illnesses. So maybe the next E. coli spinach scare can be stopped by a drug like BV. Medically, BV has so many possible applications for patients susceptible to nosocomial infection, such as burn patients who are at a high risk of colonizing Pseudomonas in their wounds, and catheterized patients or immunocompromised patients who are susceptible to pathogenic E. coli. The antibiotic resistance in these strains is the highest amongst infections, and new tactics of treating them are needed soon. So, where did this bacteria come from? Well, it was actually discovered by accident in soil samples, but it's also present in the gills of fish and other aquatic creatures, and it was recently shown to be in mammalian feces, indicating its presence in the digestive tract of humans. So, this sounds pretty good, right? What about some criticisms? Will resistance be a problem? So probably not, as there's only one case of this resistance happening in similar organisms, never reported in this organism, and that's probably due to low levels of recombination and low levels of horizontal gene transfer due to the lack of plasmids within BV itself. So what about an immune response since it is a bacteria? So it actually has a 100-fold decreased immune response in comparison to E. coli, which is most likely because the phosphates normally found in the lipopolysaccharide outer layer are replaced with alpha-D mannose in BV, which make them have a lower binding affinity to immune cells and less of a reaction. But is it still effective? So technically it doesn't kill all of the cells, but it does kill every bacterium that it does invade. But it's highly resistant to beta-lactam antibiotics, so the potential for dual therapy is there. Can we culture it? Not on traditional agar, but we can put it in a solution and culture it there so long as it has prey or gram-negative bacteria available for it to replicate. So, in the end, do the pros outweigh the cons? Based on what we know, I think so, but anything can happen when model systems are put to the test. So we really need more research to know for sure. But given the current trend in antibiotic resistance, I think this is as good of a bet as any.